Welcome everyone to this week's edition of the Extracellular Vesicle Club. It's my special pleasure to introduce our guest today, and specifically I will introduce Lei Guan, who is at University of Pennsylvania, a very valued colleague who's published some seminal work in the field of extracellular vesicles and cancer. And Wei, thank you so much for offering to share some of your recent research with us today. I'd like to hand this over to you to introduce our presenter.
All right, thanks, Ken. I will be very brief. Lei was a former postdoc in my lab at the University of Pennsylvania. Lei currently is an investigator at the Xijing Hospital in China. Lei is a really talented young scientist who is, or say, innovative in thinking and meticulous at the bench. So I just wanted to add that besides EV, Lei loves fishing. He not only has the knowledge of different kinds of fishes from all different types of waters, but can also deliver a fish dish that will probably will enchant any food critic. So Lei probably have a lot to show to today. So without further ado, Lei, you can start.
Okay. Hi, everyone. I'm Lei Guan from Weiguo's lab. Thanks for Ken giving me this opportunity to introduce our work about the tumor-derived immunosuppressive EVs and CD8 infiltration. So, okay, let's begin. So, as we know that the endosome sorting complex required for transport escort machinery play an important role in exosome biogenesis. This figure shows the fourth subcomplex of escort machinery. HRS is a key component of escort as it mediates the initial cargo recognition and sorting into endosomes. HRS binds to ubiquitinated cargoes through its ubiquitin interaction motif. So as HRS plays an important role in exosome biogenesis, we screen the potential phosphorylation of HRS by oncogenetic kinase in cancer cells. Using mass spec, we analyze HRS purified from the melanoma cell line and identify a phosphopeptide in which serine 345 was phosphorylated. To test whether ERK phosphorylates HRS in vitro, we express the HRS in HEC293T cells, then purify HRS from cell lysis and treated with constitutively ERK, active ERK, or kinase-dyed ERK. This CA ERK, but not KD ERK, phosphorylated HRS, suggesting ERK phosphorylate HRS directly. To verify the phosphorylation of HRS by ERK on serine 345, we mutated this serine residue to adenine, then purify HRS. This point mutation abolished the ability of HRS to be recognized by anti-ERK phosphosubstrate antibody. As a control, mutating the adjacent serine residue did not abolish HRS phosphorylation. So in cells, HRS phosphorylation is increased in response to EGF treatment, which is known to induce ERK activation. Treatment of cells with ERK inhibitor block HRS phosphorylation. So next, we analyze the protein composition of the SEVs derived from cells expressing different HRS mutants. We found that PDL1 was identified in a group of membrane proteins that there were most significantly enriched in SEVs derived from phosphomimetic D mutant as compared to the phosphodeficient A mutant. So our previous work shows that HRS knockdown can inhibit exosomal PDL1 secretion. And also HRS can bind to PDL1. So this IP test shows that PDL1 interact with strongly with D mutant, while the interaction was diminished by the A mutant. Neither mutation affected the interaction with HRS bound with E-cadherin or STEM, a known partner of HRS in the escort subcomplex. Our domain mapping experiments show that HRS lacking UIM domain retain PD-L1 binding, but whereas further deletion of serine 345 abolished the interaction between the HRS and the PD-L1. So this data suggests that the serine 345 phosphorylation related to ubiquitin-independent cargo sorting compared with the wild-type HRS. PD-L1 shows the significantly decreased colocalization with A mutant and the increased colocalization with D mutant. So this nanoparticle tracking analysis shows that there was no difference between the total number of EEVs secreted by these mutant cell lines. So we next test whether the surface PD-L1 or the total PD-L1 protein levels still show no change between these mutation cell lines. So so for the EV protein levels, we examined the level of PD-L1 was significantly between these cell lines. We find that the amount of PD-L1 was significantly reduced in SEV from A mutant and the increase in SEV from D mutant. So there's also detected in the human and mouse cell lines. So we also examined the ERK inhibitor on SEV protein PD-L1 expression. This BVD523 also work at the ERK inhibitor. So BVD523 inhibit the HRS phosphorylation and reduce the loading of PD-L1 on SEVs. So in cells express HRS wild type and BVD523 treatment decrease the amount of PD-L1 in EVs. In contrast, BVD523 did not change the level of PD-L1 in EVs derived from D mutant. So based on HRS sequence, we generate an antibody to detect the PHRS. So you can detect the HRS phosphorylation at serine 345 efficiently. We find that using this antibody, we found that the distribution of PHRS and CD8 T cells in tumors tissues was examined on the melanoma patient TMA. So we use this microarray. So from the result we showed, we know that there was lower level of CD8 T cells in tumors with high levels of PHRS compared to tumors with low PHRS. The number of CD8 T cells were inversely correlated with the level of PHRS in primary melanomas. So as a control, there was no correlation of CD8 T cells with the level of total HRS proteins in melanoma. So in the next step, we also tested the lymph node metastasis melanoma. So we find a similar correlation between CD8 and phospho HRS. So we know that the human individual tumors have a heterogeneous expression of kinase and CD8 infiltration conditions. So when we test the same patient, we find that the PHRS was heterogeneously expressed, and a significantly low level of CD8 T cells was observed in region of tumors with high infiltration of CD8 T cells. So using this computer imaging analysis, we find that CD8 T cells was greatly diminished in the areas with high PHRS. So how about at the tumor stroma border? We find that fewer CD8 T cells migrate into phospho-HRS high region. But together, these results suggest that HRS phosphorylation is associated with spatial restriction of CD8 T cells in melanoma. So CD8 T cell infiltration is associated with anti-tumor immunity. To investigate whether HRS phosphorylation in tumors involved in immunosuppression, we established stable Yammer cell lines that express wild type or phosphodeficient A mutant or phosphomimetic D mutant HRS. So D mutant tumors grow significantly faster than wild type and A tumors in wild type mice, but not in tumor deficient RAC2 knockout mice. So the flow cytometry also shows the same result, that the number of PD-1 positive CD8 T cells was significantly lower in D mutant. The amount of CD8 T cells is expressing Ki67 or greater than B was lower in wild type and D mutant tumors. So the antibody, the anti-PD-1 antibody efficiently inhibited the growth of the tumor expressing A mutant. The inhibitory effect of PD-1 blockade was decreased in wild type tumors and nearly abolished in D mutant tumors. So these results suggested that the immunosuppressive induced by HRS phosphorylation inhibit the effect of PD-1 blockade, and raise the possibility that the inhibition of HRS phosphorylation would enhance the effect of PD-1 blockade. To test this possibility, we combine the anti-PD-1 blockade and the ERK. To test this possibility, we combine the anti-PD-1 antibody and ERK inhibitor BVD523 in B16 tumors, which are known to be refractory to PD-1 blockade. So this B16 F10 tumors harboring different HRS variants did not show significant difference in tumor. So in PD-L1 knockout B16 tumors, in the PD-L1 knockout B16 tumors, the D mutant failed to suppress CD8 T cells, suggesting an important role of PD-L1 in HRS. So in the next slide, we saw we examined the effect of SEVs from different HRS mutant cells on tumor progression. So infusion of wild type SEV or D mutant SEVs derived from B16 cells expressing PD-L1. So this SEVs significantly promote PD-L1 knockout B16 tumor growth in wild type mice. So the effect was not observed in RAC2 knockout mice. So we also tested the infiltration of this CD8 T cells. We find that this the total number of CD8 T cells and the Ki67 and the greater than B percentage was suppressed by the wild type and the D mutant, but there is no difference between the control and the A mutant. So to examine the direct effect of these EVs on CD8 T cells in vitro experiment, the wild type and the D mutant SEVs, tumor-derived SEVs, also show the same significantly inhibit T cells proliferation and activation. Also we use the RPPA to detect which changed in the T cells. So we find that there is a cluster of proteins related to T cell proliferation and activation, such as FOXM1 and RA and ASNS or cyclin B1, also show the decrease in wild type and D mutant SEV treated T cells. To examine the function of ECM bound SEVs on T cell migration, we use an in vitro in vitro transmigratory assays. These transfer wells were coated with a mixture of SEVs and fibronectin to mimic the ECM loaded with tumor-derived SEVs. So the transmigration of these T cells was greatly reduced by the ECM loaded with Y type and the D mutant SEV. So then we examined the cytotoxicity of T cells. So the mouse splenocytes treated with Y type and the D mutant EVs showed reduced cytotoxicity against B16 and F10 cells compared to treatment with PBS or the SEVs derived from A mutant. So previous observation raised the possibility that these exosomes associated with ECM surrounding tumors block T cell infiltration. So here we transfer the CD8 T cells into RAC2 knockout mice and inject the PD-L1 knockout B16 cells. So after that, we mix these cells with SEVs and matrix gels, which is often used to mimic the ECM. So we found that the infiltration of transferred CD8 T cells was inhibited by the Y type and D mutant SEV. So since the PD-L1 was enriched in the exosomes but not altered on the cell surface as a result of HRS phosphorylation. So we examined the role of SEV PD-L1 in PHRS-induced CD8 T cell suppression. So from results, we can see that the Y type and D mutant SEV derived from PD-L1 knockout B16 cells did not show any inhibitory effect on the cytotoxicity of CD8 T cells. So pre-treatment with anti-PD-1 blocking antibodies attenuated their inhibitory effect on CD8 T cells. So these mouse EV infusion experiments show that PD-L1 blockade on SEVs attenuated their tumor growth and the promoting effect of an inhibition of CD8 T cells by Y type and the D mutant SEVs. So we also detect this CD8 T cell infiltration rate. So to specifically analyze the role of PD-L1 on SEV's inhibitory effect in vivo, the amount of tumor-infiltrated CD4/5 protein 1 CD8 positive T cells treated with Y type or the D mutant SEVs were significantly lower. As the enrichment of PD-L1 on the SEVs can be blocked by ERK inhibitor. So we examined whether BVD 523 treatment of tumors can modulate the inhibitory effect of SEVs. So these SEVs from cells treated with BVD 523 lost their inhibitory effect on T cell migration and function and cytotoxicity, and so there was no difference between control of BVD 523 treatment for D mutant SEV. So here is our working model. We show that HRS phosphorylation drives immunosuppressive exosomes secretion and restrict T cells infiltration into tumors. So these high PHRS levels give immunoreactive armors to these tumors and reject these CD8 T cells infiltration. So here come to the conclusion part that HRS key players in exosome biogenesis is a direct downstream phosphosubstrate of ERK in metastatic melanoma. So HRS phosphorylation promotes the selective loading of PD-L1 to exosomes, which suppress the proliferation and the function of CD8 T cells and block their tumor infiltration. So phospho-HRS IHC combined with the assay of exosomal PD-L1 in plasma may provide important diagnostic information that will help us to identify this patient who will be likely to benefit from anti-PD-1 treatment. So acknowledgement. So first I would thank my PI, Wei Guo, gave me a lot of instructions and time to finish this work. And I also would like to thank all of my collaborators, especially like Bin Wu from our lab, and the Ting Li, and from the Penn Medicine, and Xiaowei Xu gave us a lot of suggestions and sample analysis help about the melanoma patient. And Lin Beer and David Spector give us a lot of help on protein mass spec data analysis and processing. And also the Fox Chase Cancer Center, Casey, and the Fangping give us a lot of help on patient sample analysis. And the NAPES person who named Guangcan Shao and their PI Meng Qiu gave us a lot of help on the phospho HRS site identification. And other people like the Golden Mills gave us a lot of help on the RPPA mass spec, and KC Flaherty gave us a lot of help on the clinical data analysis. So that's all about my presentation.
Well, thank you so much, Lei, for sharing this with us. And I just want to say that if your fish dish that we heard about from Professor Guo is anywhere near as good as your presentation, then I think you're going to have a lot of people knocking on your door tonight. So thanks again for this. And before we get to the questions, I just want to remind everyone, including those of you who may have joined during the presentation, to just put your questions in the chat box, please. And then I will get to you. You are also able to now unmute yourselves and show your video if you would like to. So I would just like to start with a question of my own, and this has to do with what I think is a big question in the EV field right now, and that is exactly how EVs are exerting their influence. So I think you've shown very clearly that you have this surface display of a molecule that is happening. Do you feel that this is the primary means by which your EVs are exerting their effect, or do you think that there is also a cargo delivery aspect here that might also be important?
So for this project, at the very beginning, that our previous work, like Gandhi, showed that when these EVs interact with T cells, so we take a long time to observe these phenomena. We find that for CD8 T cells, almost all these EVs cannot phagocytosis by these CD8 T cells. But we also sure about that like the macrophage of these, they have a very strong ability about the phagocytosis these EVs. So in that case, this kind of EVs may deliver a lot of cargoes transferred to the target cells. Yeah, but we're not involved in that phenomenon in this project.
Sure.
Yeah.
Okay. Yeah, and I also found it very, very interesting that you were using both human and mouse cells, and you were seeing that the same effect was being exerted by both by I think it was the WM9 cells, and I can't remember the mouse line, but that was also quite interesting to me. All right, so we do have let's see here. We have a question coming in the chat box from Qingling Fu. So Qingling, nice to see you. Feel free to ask your question.
Nice to see you, Ken.
Hi.
Hi. My question is, how about the potential clinical application of blocking HRS or HRS-rich SEV, and what will you do next? How to block this pathway and to increase the CD8 T cells to responses to PD-L1, to anti-PD-L1.
Yeah, so from the let me show the. Yeah, so in the human samples, we find that there are seems a lot of research focus on how to improve the react of the ICB. So in our case, we find that the high PHRs seems to restrict the CD8 T cells. So we know that the ERK inhibitor already in clinical trial raise a lot of questions that because this ERK is very essential for every kind of cells. So they may show more more toxicity to all these healthy cells. So we may think that this PHRs, if we can inhibit this phosphorylation very specifically, we may have a more safer and effective effect to improve the SAB survey. Yeah.
Okay. Maybe you can use some treatment to inhibit the production of SEV. Yeah, because you mean that the HRS is because it can be inhibit the T cells responses to anti-PD-L1 because in SEV. Can you try to some treatment or some treatment to block the production of SEV? Yeah.
Yeah. So as we now all know that, like the knockdown of Rev27A or this process, also I saw some drugs can use to inhibit the EV production. But I think the question is that how to specifically use these drugs to the tumor cells versus other cells. Yeah. So
yes,
maybe yeah. So that's why if we totally block the EV secretion, they may cause a lot of problems on the healthy cells. Yeah. So
okay,
that's
maybe
why
local application for this medicine or this drugs or the others. Yeah.
Yeah. So it's a very good question. Maybe we can test this in future. Yeah.
Okay.
Whether
very
they have
perfect
better
work. Thank you.
Yeah. Thank you.
Yeah, thanks very much for the insightful questions. And I think related to that, I'd like to ask about the reach of this phenomenon. How how do you think this is happening throughout the body? How far away from the tumor, or can you estimate this? Is it simply that you're talking about the T cells that are they're trying to get into the tumor, or is this something that is changing with regards to the quality of the cells throughout the organism?
I think that so in our cases that I think that the EVs, if they have a very strong effect, they need maybe combined with a lot of high concentration EVs in some area. Yeah. So it's according to the dose of the EVs and the strongest of these EVs. So maybe we can detect the EVs in the plasma, but maybe they can reflect the total aspect of these tumors. Yeah. But when they talk about the function, maybe they need a lot of conditions to function well. Yeah.
Very good. All right. Well, let's next go to Natalie Turner.
Hi, thanks for the great presentation. So just a disclaimer, I am not a cancer biologist by any stretch of the imagination, so I apologize if my question is a little bit silly. But I just wondered, I guess targeting the phosphorylation pathway is going to be very challenging because it's not just in the tumor cells, it's obviously systemic in lots of pathways. So my question was really related to, would it be more feasible to load synthetic nanoparticles with the non-phosphorylated HRS and deliver that directly to the tumor if HRS is in fact beneficial when it's not phosphorylated. And if that's completely silly, then would you consider loading synthetic nanoparticles with something that might be beneficial for tumor treatment? As melanoma is obviously accessible, it's a skin cancer, it would be kind of feasible that you could deliver something directly to the tumor.
Yeah, first, thanks for your question. Yeah, it's a very good question. And for the clinical purpose to reverse this phosphorylation, maybe we can use nanoparticles to deliver the phosphodeficient HRS as a therapeutic drugs to lower the PD-L1 exosome enrichment on the SEVs. Yeah, so if the nanoparticle works very well, so it will be very promising to try this on the future experiment. Yeah.
Great, thank you.
Thanks so much, Natalie. We have a question now from Lin Li from Southeast University in China.
Thank you. I'm actually a nephrologist, so I'm trying to ask a question about the methodology. Could you please explain more about how to mimic the behavior of EV in ECM in your study? I'm interested in this part. How to track or observe EV in ECM, such as any difference regarding their behavior in the ECM, like the movement of those EVs in ECM. So I'm not sure. Could you please explain more about the methodology? Thank you.
You mean how? Thanks for the question. That you mean how we monitor the EV migration in this ECM?
Yes. And
yeah,
how to observe them in the ECM?
You mean in vivo or in vitro? So the EV migration?
I think in maybe in your study you use in the transwell study.
Yeah. We use it here. Yeah.
Yes.
Yeah. So you mean the method to detect the other cells interact with ECM bounded EVs, or we just monitor these EVs?
Okay, you just mix the EV with ECM together and
yeah.
Okay.
Yeah, because that when we isolate some EVs from those xenographs or allografts, we find that some EVs can show the like the bond with fibronectin or something ECM. They show like the pore on the chain. So we propose that they may bind together, and because I think in the ECM it's not totally like the liquid condition, so the EV can move everywhere. Maybe they can interact with ECMs and they can hold it there and give a high concentration. But I think for the EVs migration, or they may diffuse because of the blood vessels goes here, and they may leak some from the ECM, and we can detect in the plasma. But for the ECM bounded EVs, I think it's quite tightly between the EVs and this ECM. Also, if we know that if these EVs want to move around here or move to other place, maybe they need a lot of energy. So for the EVs, I think it's maybe quite small. They do not have enough enzyme to give them enough energy do this migration. Yeah.
May I add something? I actually regretted I asked Lei to cut off some slides because it would be too overwhelming. But one slide he want to show actually was included in the paper is that he showed the EM of EVs associated with the ECM fiber. It's almost like a tomatoes on the vine. It's a really organized structure. And David Leiden work showed that the EVs actually carry integrin, which bind to collagen, I forgot the collagen or fibronectin, bind to ECM. So ECM definitely help. Well, I think very likely ECM help organize the EVs in the tumor microenvironment. It's almost like a landmine deployed there, so prevent T cell from entering. That also relate to Ken's question, or someone else. Indeed in the EV, the T cell actually, if you see Lei's picture, the IHC picture, the T cells actually get to the border, but they just cannot get in. You can see right here. Yeah, you can see right at the border. It's almost like they cannot step into the landmine. Yeah.
So that's very interesting.
Yeah.
Go ahead, please.
Maybe the different characteristics of the ECM would have some effect on the migration of EV.
Could be. For example, in pancreatic cancer, there's very dense ECM, and I think the organization of EVs on that tumor microenvironment could be different from other types of tumor microenvironment, such as melanoma. Yeah.
Okay, thank you very much.
Thank you, Lin-Li, for the questions.
It's a good question. Yeah.
Yeah, I think on that same note, I'm reminded of this paper that came out just a little bit ago in Nature from Weems et al., and that was on melanoma blebs. And of course, they were looking at something very different about how the blebs are affecting the cell that's making them.
Right.
But that paper also led me to wonder about EVs, larger EVs, because it's something that buds from the cell surface that can end up as an EV, but as probably a large one. Do you observe any of these large EVs in the tumor microenvironment, or are they mostly smaller ones? And I mean, I'm wondering what would, because if you have a very large EV, that could actually still contain cytoskeletal components, mitochondria, it can even move around. Do you think that larger EVs could have similar effects to what you're seeing with these small EVs?
Yeah, so I think thanks. It's a very interesting question. Yeah, so let me separate into in vivo and in vitro systems. So the in vitro system, when we culture these cells, when we find these cells in very good condition, they show less produce on these big EVs or macrovesicles, because these macrovesicle may when these cells shedding these big macrovesicles, they may need more energy, like the human lost their arms, lost their legs. It's harms to these cells. So when we culture these cells with a good FBS, or it's always showing good condition, so they also show they have high production on these small EVs, like these exosomes. So, but for the in vivo condition, I think that because a lot of T cells, they may killing these cells. So when they killing these cells, these cancer cells may show a lot of not only EVs, they may also show autophagosome and apoptotic bodies, or these cancer cells will disassociated with these large EVs, very, very large EVs. But I think these are quite big, large EVs also show a very strong effect on these immune cells, like because like for the macrophage, for the certain macrophages, they may have the certain ability to catalyze to accept these big vesicles. But these are a lot of big vesicles on the border or on the ECM. They will show a very big burden to these immune cells. Maybe they show very strong effect. Yeah, so they may not this problem for FEVs. They may show a lot of function when after these tumor cells dead or necrosis or apoptosis induced by these immune cells. Yeah, they may show a very big burden to these immune cells. Yeah.
Very good. We have another question here from Jeff Buffard. Jeff, please go ahead.
Yeah, sure. It's late here, so I'm going to leave my camera off. But so these EVs bound to the ECM.
Hey, no fair, no fair. I've got mine on. No, go ahead.
Are they taken up by the T cells and removed from the ECM fibers, or do they stay there and kind of decorate and then continue releasing signals?
You mean this EVs bind to the ECMs?
Yeah. So these tomatoes on the vine.
Yeah.
Do they stay there, or do the T cells pick them off?
Yeah. So so we observe the TDEVs, those tumor tissue derived EVs, how they work with T cells. Maybe I think when I will show a little picture. Give previous work that when we detect these EVs bind to these CD8 T cells, right? So we see this bind will exist a very long time, maybe more than 48 hours. This EVs very tightly bind to the CD8 T cells. So I think when the T cells enter the tumor border, they will trapped by these EVs and also bind with the fibronectin or some ECMs outside the tumors. So I think maybe this a very big trap. So that this EV is like the clap to restrict these cells, cannot migrate into these tumors. Yeah.
Yeah, so also I think it's a very reasonable explanation. Of course, it's a speculation at this stage, but also relate to Ken's first question. Cargo delivery internalization versus surface inhibition. There are two modes of interaction between EV and the target cell. I think it reminds me of this paper from Teresa Whiteside from University of Pittsburgh many years ago. She actually looked at the internalization of EVs by different immune cells, and she found that the CD8 T cell actually is very bad at internalize those EVs, although the binding is okay. So this is our observation seems to be consistent with her data, where we see the surface attachment of EV for many hours, days, but not internalization. So that I think this probably one speculation is that probably the inhibitory effect delivered by exosomal PD-L1, for example, already exhaust or make this T cell dysfunctional. So they're maybe not good at internalization. That's just my speculation.
That's super interesting. Thank you.
Yeah, so it also reminds me a recent paper that they target the ECM, just target the ECM to improve the ICB response. So maybe I think only if they target the ECM, they may move out the physical barrier between these tumors and these immune cells. But for this case, we may propose that there are also some chemical barrier between these tumors and the immune cells, not only the ECM. Yeah.
That's cool. Thank you.
Yes, very cool indeed. I think it's fascinating to think about the EV as a durable signaling platform, where it's not just this one time and you're done kind of delivery of cargo, but you could actually have something that's sticking there for 48 hours and might be bumping into multiple cells. Very interesting and very efficient of the tumor to do that, perhaps. So it looks like we have reached the end of the questions in the chat box. If anybody has a last question, please put up your hand now or put something in the chat box quickly. But if not, we've heard about fish tonight. We've heard about some tomatoes. I'm getting a little bit hungry, so but thanks for sharing this very exciting work and I think very consequential work with us. And we wish you, Lei, and also you, Wei, all the best as you continue to bring good EV research to us. So thanks also to everybody who's joined. It's great to see all of the participation, especially I see a lot of people from the Asia Pacific region today. It's morning for you, it's evening here in the Americas, but from wherever you are, thanks so much for contributing. And again, a special thanks to our presenters. And we look forward to seeing everyone at a future edition of the Extracellular Vesicle Club. Thank you all, and bye now.
Thanks, Ken.
Bye.
Thank you.